Hi everyone, welcome back. In the previous lecture, we've learned the basic operation in R and the work environment RStudio. Now let's start with the CummeRbund and learn how to use it for the downstream RNA-seq data analysis. The most advanced feature of CummeRbund is that it takes output files from a CuffDiff run and creates an SQLite database of the results, describing appropriate relationships between genes, transcripts, transcription starting sites, and the CDS regions. Once stored and indexed, data for these features, even across multiple samples and conditions, can be retrieved and allows the user to explore the subfeatures of individual genes or gene sets. In other words, we are flexible to create our own sets of genes and analyze their relationships using the RNA-seq data. These are the steps we are going to demonstrate. Now let's get back to RStudio and load the CummeRbund package. So first, let's go back to RStudio. I've prepared a script for today's tutorial, so I'm gonna go ahead and open it first. Let's take a quick view of the script. First, we will create a database from the output of CuffDiff, and then we will perform some basic analysis, and the last we will perform a hierarchical clustering using the output data. First, we need to upload the CummeRbund library. So simply use the library command. As we can see, CummeRbund uploads itself as well as some other packages it needs to use. The next step is to set up the working directory. It gives me some error, so I need to change the path. Here I can copy paste the path to the set working directory function. And this error is a typical error you will encounter in the Windows system. The solution is to use double backward slash instead of a single one. The single backward slash should be working on Linux and Mac system. And these two steps are probably the same for all our tasks. First, upload your library, and second, set your working directory. So now we will create a CummeRbund database from the CuffDiff output. Remember, one of the advantages that CummeRbund offers is that we can ignore the individual output file from CuffDiff, and as long as we create a database using CummeRbund, it will take care of all these output files at once, and we don't need to worry about the individual relationships. So we will create a database using the readCufflinks function. Let's first create a database for the G1 time point. So as soon as we execute this readCufflinks function, under the console we can see the information or the progress. We can see it's writing the TSS table, the coding region table, etc. Alright, now we have our first CuffDiff database called CuffData, and the type of this variable is a CuffSet. What is a CuffSet? Let's go to the package tab and click on CummeRbund, and the whole help documentation will show up for CummeRbund. And here we can check the information for the methods and classes. Here is the CuffSet class. You can read through some more details about CuffSet, but we don't need to worry about for now. Now first, I want to plot the distribution of the expression levels for each of the samples. What I need is the CS density function. And since we are we want to see the expression level at gene level. So we will first extract the information at the gene level from this cuff data database and then plot the density distribution. This step is in general pretty fast, and as soon as we type the variable, we can see the figures under the plots tab. So we have different colors for wild type and knockout, and we can see that the majority of the genes, the expression of the majority of the genes are within the same magnitude. This is what we want to see. Next, I want to compare the expression of each gene in the two conditions with a scatter plot. And we can do this by using the CS scatter function. Again, we want to plot at the genes level, so we will first extract the information based on genes level. And we also need to specify which two conditions are we looking at. And we can pass these two conditions as an argument in the CS scatter function. Be careful that the orders matter, since CS scatter will put the first argument, the first condition as the x-axis and the second condition as the y-axis. So now I will go ahead and plot the scatter plot for each of the genes. This step is quite time consuming. So I will skip the running time and show you the screenshot of the actual plot. So here we can see we have the wild type as the x-axis and knockout as the y-axis, and from the scatter plot we can see that most of the genes are well correlated in these two conditions. However, if we look carefully, we can see that genes located here are relatively higher expressed in wild type but lowly expressed in wild type in knockout. And on the other side, we have some genes that are highly expressed in knockout but lowly expressed in wild type. And these are the genes that we might be interested in because we are identifying the differentially expressed genes. So how do we tell if this difference is significant or not? Well, we can do the volcano plot. So I'm gonna call this CS volcano function and specify the genes that I wanna specify as the gene level. So here is the volcano plot. On the x-axis, we have the log2 of the fold change of each gene in the two conditions, and on the y-axis, we have the log10 of the p-value. So basically, the upper the data points locate, the more significant the change is. And we also have the legend for the significant. The black data points represent the genes that are not significantly changed, while if we look up here on top, the red data points are the ones that are significantly changed between the two conditions. If we look at the data points that are located on the left of the 0, they are downregulated in knockout, where the ones on the right are upregulated in the knockout. And if we're happy with this figure, we can export it as either a PDF file or export it as an image. And R has offered many different formats for the image, from the basic PNG, JPEG, until the vector graph like SVG and EPS. And if we export it as this vector format, we can further edit it in the Illustrator, and it will increase the quality of the figures. So next, what if we have a gene that are particularly interest to us? We can plot the expression level of a particular gene by using the expression bar plot. So first we need to extract the information associated with this gene from the database by using the getGene function. and we give the value to my gene. Now let's look at my gene. We have all the information associated with this gene BRCA1. We have some different slots, for example, annotations, FPKM, or the read counts. So now I want to go ahead and plot the expression. Cufflinks use the FPKM value as the default measurement for gene expression. So if I zoom this figure here, I will see the expression for BRCA1 in knockout on the left, wild type on the left, and knockout on the right. And we can also see the FPKM value from the y-axis. Except for the expression, we can also plot the isoforms for this gene. Here it doesn't change. It's probably BRCA1 doesn't have isoforms. Alright, so these are some basic analysis we can do using CummeRbund. If we go to the menu of CummeRbund, it provides a very comprehensive example of what you can do with CummeRbund. For example, we can do the dispersion plot, we can do the SCV plot, and scroll down. They also show some example of the pairwise scatter plot. So here we have more than 2 samples, and we can plot the scatter of the pairwise condition. And the individual scatter plot. We can also do the clustering of the samples based on their gene expression profiles. MA plot and some pairwise volcano plot. And here is a list of examples of how to manipulate your data. And if you scroll down here, we can see the relationship between different tables stored in the database. If you're an advanced user, you can make use of this relationship to customize your analysis. Alright, next I want to show you how to perform a basic hierarchical clustering of differentially expressed genes in the 3 conditions. So remember we have 3 time points, G1, G2, and G3. So first I'm going to create a database for each of the conditions, and then extract the gene expression level from these 3 using a pre-made gene list. And then I will merge the matrix from these 3 into 1 and perform the hierarchical clustering using the merged matrix. So within each of the condition, we can identify a list of differentially expressed genes. And I've prepared a gene list where genes in this list are at least differentially expressed in one of the conditions. We can take a look at the gene list. It's basically just a simple list of genes. So I will read this gene list and store into a variable, and then using this variable to extract all the information associated with the genes listed in this variable. So first, let's create the database for each of these conditions. I've already have G1 created. It'll start from G2 and goes to G3. So similarly, we can see that CummeRbund is writing the tables for genes, for TSS, for isoforms and coding regions. And now it moves to G3. Okay, now we have, next I want to upload the library called gplots. It's the one that will perform the hierarchical clustering. We can upload it here. We can also click the checkbox under the packages list. Next step, I'd like to read the gene list from this file I've already prepared. So here I'm using the read delim function, and now as we can see from the workspace, we have almost 2200 genes. So next I'm gonna extract the expression information that associated with the genes from each of the 3 databases. Since I will plot the hierarchical clustering based on the FPKM value, I will use this FPKM matrix function to create the matrix. So this is a compact command. It equals to these 2. So first we will get the genes from the database according to our gene list, and then so get genes will give us all the information that associated with the gene with the list of gene. But what we really need is this FPKM value. So I will further extract the FPKM value of this gene list, and I will assign this FPKM matrix to a variable called gene1.matrix, and I can repeat this step for G2 and G3. And here is the progress. So now I have 3 matrices here. Each one has the same number of genes, and we can take a look at of the first few lines for example, gene1.matrix. We can see that each gene represent a row, and the 2 columns correspond to wild type and knockout. So next I'm gonna merge these 3 into one matrix, and we can do this by using the Cbind function. Now if we look at the first few lines of this merged matrix, we can see that the rows stay the same as each gene, and we have 6 conditions, wild type and knockout, in each of the time points. Now we can finally perform the hierarchical clustering. I suggest you to use the heatmap.to function. It's an improved version for the heatmap function. So first we will need to convert whatever type is our merged matrix into a matrix, and then using the scale equals row to specify that we want to normalize across the rows, and we want to show the dendrogram on both rows and columns, and we can hide the density information from now, and we can keep these, and these are some parameters that are not that important, but for this lab row, it's the label of rows. So since we have over 2,000 genes, it wouldn't be visible for the labels to show on the plot, so I set up as false. And as soon as we execute it, R will start perform the analysis, but this step is quite time consuming. So again, I'll show you the screenshot of the result. So here is the hierarchical clustering plot we can get. We can see that the columns are the conditions, and we can see the dendrograms on both rows and columns. We also have the color key on the upper left corner, which indicates the normalized z-score. So remember, we normalized the expression value across the rows, and now the red color represents a lowly expressed gene, and the white color represents highly expressed genes. Now, one of the things that so there are a lot more parameters that we can play with in the for the heatmap.to function. So if we search it in the help, we can see a full list of parameters that we can set. Two most important parameters are the distance function and the linkage function. And you can read more about these two under this help documentations. It has some links to jump to the distance functions and linkage functions. So one thing I want to point out is that sometimes the color scheme that heat map function used by default is not very distinguishable. So I found this very good color package that we can use very easily to change the color scheme for the plotting. Down here, it's called R Color Brewer. So basically, we just need to specify that we don't want to use the default color scheme. We want to use the color that offered by this brewer package. Now we can see that the color scheme has changed, but the shape and the structure of the dendrogram, the clustering is not changed. It's only the color that represents the value changed.
Besides hierarchical clustering, we can also apply other downstream analysis approaches to the RNA-seq data. For example, the principal component analysis is usually used to identify groups of samples showing similar expression profiles. Pathway analysis is able to detect enriched pathways given a list of differentially expressed genes. Enrichment analysis is similar as pathway analysis. It's expanded to identify other enriched properties for the list of genes, such as a biological process, molecular function, or transcription factors. We can also seed the differentially expressed genes in a biological network and explore the relationship of those genes based on the physical or functional links.